Hey, and thanks, Bruce, for the introduction. So as he said, I'm going to talk today about a few different emerging technologies for managing N2O nitrous oxide production from corn agriculture. And so this is some work that I was working on in my postdoc at the University of Minnesota with Tim Griffiths, as well as some other collaborators from the USDA Agricultural Research Service. So a little bit of outline about what I'm going to talk about today. So first, I'm just going to briefly overview why we're interested in N2O and atmospheric loss of nitrogen. And then I'm going to talk about two different technologies and their potential for actually reducing nitrous oxide emission from agriculture. So the first one will be procyanidin amendments, and the second one will be Pivot Bio's proven product. So why do we care about the atmospheric loss of nitrogen? So this figure is just a very oversimplification of why the economic reason why we care about the loss of nitrogen. So are we getting quieter? Okay. Sorry if I was too loud. Our farmers apply nitrogen so they can have higher yields in crop growth. Some of that nitrogen is lost to the atmosphere and essentially comes out of our pockets. So if we're applying nitrogen that ends up in the atmosphere, that is money that's unrecovered. We would much rather that this picture looks like this, where we can apply our nitrogen, our yields are higher, and we have less loss of atmospheric nitrogen. And so, although it's variable, so as we've heard in some of our earlier speakers, the amount of loss of nitrogen to different pathways of the atmosphere are very dependent on your soil type and your weather. It can be as high as, for example, 40%, usually not quite that high, but it can be quite a significant portion of your nitrogen budget. In addition to the economic reasons, we also care about nitrogen loss due to its negative environmental effects. So many of us are applying nitrogen as farmers here on fertilizers, the left, and then there's transformation and transport kind of on the daily to seasonal scale to different atmospheric reservoirs as well as to aquatic reservoirs. So we have things like ammonia going to the atmosphere, contributing to air pollution, as well as nitrates and ammonium to water pollution and groundwater pollution. And then what we're particularly focused on here is this N2O, nitrous oxide pollution, which, as we look on these longer term impacts, that eventually contributes to climate change. And as we heard earlier as well, is about 300 times more potent on a mass per mass basis as carbon dioxide. We think about climate change. So quite important on a longer term scale. We're also worried about these other effects of nitrogen, for example, eutrophication going to our waters, or in biodiversity loss as we fertilize non-agricultural ecosystems with atmospheric deposition. And so N2O, it's no laughing matter. So many of you likely know that nitrous oxide is also known as laughing gas. So it's my little pun for the day. But it's something that's quite pressing and a challenge for us as we increase agricultural production. So on this left here, we have a map of nitrogen fertilizer use in the US between 2000 and 2015. And you can see, it's a little bit difficult to see here, but we're moving from kind of an orange color to a red color in southern Minnesota, northern Iowa. So fertilizer use is going up. And then on the right here, we have some tall tower data, which is from the Griffiths group, so the group that I was part of, observations over the last 10 years. And you can see kind of corresponding with some of these fertilizer increases, we've also observed nitrous oxide increases in the atmosphere. And again, this is a very long-lived atmospheric gas. So the increases we see now are going to stick around for a while. So there's a few existing N2O management tools. And as we've kind of learned, many of you likely know, some of these have varied success and are going to be very dependent on the system and the weather in a given year, in the way we manage things. So there's really a pressing need to develop some technologies that can confidently and consistently manage N2O. And so with that in mind, we're going to just discuss briefly two potential mechanisms that are at very different stages of development and understanding for managing nitrous oxide emission from our agricultural soils. So the first one I'm going to talk about is denitrification inhibitors. And so these are a pretty new kind of research topic. So it's not really in the field application scale, but really is in the laboratory study scale. And these work by actually inhibiting denitrification in the soil and disrupting nitrous oxide production. So, and there's some academic literature that I'll go into briefly that says this, but there's no really field validation of these or continuous N2O measurements that show that these are actually able to reduce nitrous oxide emissions. And then I'm going to go talk a little bit about the tailored nitrogen fixing microbial communities. And so these are a commercially available product from Pivot Bio's proven that we've been able to do some tests on and look at whether we believe these are able to reduce nitrous oxide emissions, and if so, how you would go about that. So a little bit of background here. So for these denitrification inhibitors, denitrification is simply the process of taking nitrate and moving it to nitrogen, atmospheric nitrogen. So in most of the nitrogen that goes through a denitrification process is going to end up as atmospheric nitrogen, which is, there's a lot of it in the atmosphere. It's generally considered non-reactive, but we know that some of it is emitted as nitrous oxide here. And that's what we're concerned about because it is that greenhouse gas in an ozone destroying substance. So denitrification inhibitors work simply by stopping different steps in this pathway prior to the production of nitrous oxide, so we get less nitrous oxide production in the soils. And so if we can stop these mechanisms or the microbial processes, we don't have nitrous oxide emission. Pretty simple idea, maybe a little bit more complicated in practice. So how we got into this work is we were kind of looking through the literature, thinking of what some of our next research projects were going to be. We found a few papers looking into what's called Asian knotweed here. So that's the picture on the left here. And this plant has actually been identified to release these compounds called procyanidins into soils as a kind of biological mechanism to get more nitrogen. It inhibits denitrification in soils where it is, and it's enabled it to actually be quite an invasive species in many ecosystems. Because it has this unique mechanism of kind of keeping nitrogen in the soil. We also know that these procyanidin compounds are present in high concentrations in things like grape seeds. So there's some of these agricultural products where we can potentially source some of these things. So there's, in an ideal simplified world, you could potentially take some of these compounds that we're extracting from things like grape seeds, potentially apply them to agricultural soils and reduce nitrous oxide emissions. So what was reported, this is kind of the graph that's been reported in the literature here, where we have N2O production on the left here, or N2O inhibition on the left. So the higher number here on this particular graph, the less nitrous oxide that's being produced. This one would imply that as we had higher dosages of these procyanidins, that we should have almost a complete knockdown of nitrous oxide production. So this graph is kind of what drove us to put these proposals in and to start this project. So we saw this and said, this seems quite promising. Can we reproduce it in our lab? So we're doing some work right now, and I'm going to go into at least this first data point here. We're trying to answer 3 different questions. First, can we reproduce denitrification inhibition in a laboratory setting? So can we take what's in the literature and can we do it in our own controlled circumstances, just to make sure it's not a crazy idea. So 2, can we identify economically viable sources of these inhibitors? Because if you can do it, but it's going to cost $1 million per acre, no one's going to want to do it. So can we identify either a synthetic source or a natural source? And then 3, can we observe N2O emission reductions under field similar conditions? So although there's been some laboratory work showing inhibition, saying that we can see this denitrification enzyme activity reduction in a bottle is much different than actually going and observing it continuously in soils. And so that's kind of the idea behind this project here. So what I'm really going to focus on today is this laboratory validations portion of this. So there's this first question. So how we do this is we do what's called the denitrification enzyme activity assay. So it's just simply, we put some soil in a bottle, we give it enough nitrogen, and we get enough carbon to really, and put it under an anaerobic atmosphere to theoretically allow as much denitrification as possible to happen. And so we do this in control circumstances, and we do this with exposure to these procyanidin compounds. And so we first started doing this with these kind of 2 different amendments here that are on the bottom there. What these actually are, it's commercially available wine amendments that are used to kind of fill out the body of someone who's trying to produce wine. Not or body of the wine, not the person. But these are procyanidins that are commercially available. And these were what previously had been used in the literature in that graph that I had shown you before. Unfortunately, when we started to do this, we actually saw stimulation occur. And so this is a really interesting and kind of contradictory result where we see this graph here. So this is a little bit different than the graph I showed you debefore, where we have kind of this denitrification enzyme activity on the left. So higher means we have more nitrous oxide production, and then dosage on the bottom. So these are for these two different sources. We're really seeing, as opposed to inhibition of nitrous oxide production, we actually see 200 or 300 times more nitrous oxide production from the addition of these. So this was a surprising result. We spent a decent amount of time looking into what are these sources of variability? Is it a variability with actually in the source of these compounds? Or is it something like differences in soil? So we kind of took that Lafort one, which was what they use in literature, and we applied it to a few different soils. We actually see almost the same thing. So this is a similar kind of graph here, where we have denitrification on the left, and then we have dosage on the bottom for 4 different soils. And again, we're seeing with these supposed inhibitors, we're actually seeing increases in nitrous oxide production. And so what this tells us is that things are a bit more complicated than maybe they're presented in the literature. Indoor, the sources that had been used in the literature are more variable than maybe reported. So using these kind of existing commercial sources that are available as wine amendments maybe isn't something we should be testing with. We need to kind of narrow in on some more particular chemical compounds that could potentially be these sources, as opposed to this larger class of compounds. So sorry, this isn't, at this point, I'm not giving you a silver bullet for nitrous oxide reduction in agricultural soils, but we're still working on this. So there's another postdoc that's coming in to kind of pick up this project and look at some of these in a more granular fashion to see if we can even reproduce those literature results. So we kind of thought we would be significantly further on this project at this point, but reproducing those literature results has turned out to be critical. And then kind of to all of us, it's a little bit of a lesson is, if we see something in literature, it's always nice to try to redo it ourselves, because sometimes it only happened once or twice, or the effect is maybe less pronounced than in under field conditions than it is under lab conditions. So we're going to continue to test some more purified compounds, and hopefully in a year or two, we can come back and tell you we have the silver bullet for nitrous oxide reduction. And so again, ongoing work, I kind of just went briefly into this. And so now I'm going to talk to you guys a little bit about something that's much more commercially available. So this is Pivot Bio's proven product. And so this is claimed to, their original product was that they claimed something about 25 pounds of nitrogen per acre for adding this product. Now they say it's effectively about 40 pounds of nitrogen per acre. And so this is not a fertilizer addition. It's actually adding an in-furrow kind of microbial addition. And so the proposed mechanism here is that it's simply, we have these nitrogen fixing microbes that grab this kind of stable N2 from the atmosphere, they convert it to ammonia and ammonium and deliver it straight to the roots of the plant. And so the idea here is that if we're delivering nitrogen straight to the roots of the plant, it has less of an opportunity to go through some of these other processes and end up as N2O or other reactive nitrogen species because they're just not kind of in that soil medium and not as broadly distributed in the soil medium. And so the folks from Pivot Bio will tell you that we should expect no nitrous oxide emission from these products. So, but of course, you have to test things like this. So we're really, what we'll be showing here are some tests of this product, whether or not we believe it's good for reductions in N2O. So first questions we're going to ask here are, can we use Pivot Bio's proven to reduce nitrous oxide emissions? Can we kind of show that confidently that we might be able to do this? And 2, can you reduce fertilizer, apply proven and maintain yields? Because that's really the big thing. If we can apply proven, and because in order to use proven and to reduce nitrous oxide emissions, you're going to have to reduce fertilizer application as well. And I won't get as much into that second question here because we're still waiting on some data from the most recent season, but I'll be able to talk pretty in detail about this first question. So how do we go about this? So we have a very unique facility on the Twin Cities campus that the Griffiths group and a few other groups on campus have helped build. So what it is 6 standalone soil columns that we're actually able to grow corn to maturity and to harvest. And we have control of radiation, we have control of water delivery, we have control of temperature. This is instrumented out for automated data collection, and we can actually get hourly data collection from this. So it really gives us quite high resolution data. And so we're pulling greenhouse gas data. So we're pulling nitrous oxide data, we're pulling methane data, and we're pulling carbon dioxide data. So in this facility, we're really well set up to do comparison tests of products like this, where we can say, okay, we did a treatment or a management method, and we can kind of do side-by-side comparisons in a controlled facility. So if any of you have worked on nitrous oxide, a lot of the field data is highly variable because you have variable weather conditions, or even within a field, the soil conditions are quite different. So this gives us kind of a controlled system where we can at least test the idea before moving into more of a field conditions to make sure to see if it holds true there as well. So here's just another image of it. So you can see these are about this tall soil columns. So you can have really full growth. We can get near, if everything's running correctly, we can get kind of near field yields, about 200 bushels an acre calculated from here. So it's a pretty cool facility. It's been around a little bit, but I think we really, the last few years has really been using it in kind of high frequency. And so the other thing to know here is we can do about 3 growing seasons a year. So, I mean, we're limited in space, but we can run the whole year round. So we're growing right now. Essentially, we'll probably harvest in a few weeks. So that's something you can't do in a field. So it allows us to run some of these tests on novel technologies relatively quickly without obtaining a whole lot of field space. So what did we actually do to test proven? So one thing we have to, proven would be applied with a reduced fertilizer rate if it's going to be actually lead to reduced nitrous oxide emissions. So we did what was kind of a paired gradient and then did it 3 times. So this is the rate here is 180 pounds of nitrogen per acre using urea. And that's the base rate. And then we did it at kind of reducing that. So 80, 60, and 40% with and without pivot bio. So every season we had 3 treatments with pivot bio, 3 treatments without pivot bio. And then we did this 3 seasons, repeated it for 3 seasons so we can look at the differences over the seasons. And I'd just like to show some of this data actually looks like as it's coming in. So this is some of the higher resolution N2O data that kind of exists, kind of getting these hourly nitrous oxide measurements. So, and I'm going to point out a few things here that you can see that we typically expect. So kind of show this looks similar to what you might expect to see in the field. So we have kind of this big peak at the beginning here, which we typically associate with fertilizer emissions. So that's something we expect in the field. You see when you apply fertilizer and you kind of have those kind of wetter spring soils, usually you have a peak in fertilizer emission. And then you see as that water is pulled out of the soils and it's hotter in those later summer months. So I know it says December, January, February, but you can think later season it's warmer. You have less nitrogen, you have less moisture. There's usually a lot less N2O production. So we're seeing some of these features that we would expect to see in the field in our system. It's kind of good from a validation standpoint. We also see other things that are promising. So you can kind of notice it's kind of like a porcupine. It's got nice spikes. So what those are irrigation events when we're irrigating because it doesn't rain inside. So we have to irrigate manually or automatedly. So you can actually see those results in almost every single one of these as it responds to our irrigation events. So it's a really nice data set from an N2O perspective where we can really see some of these features we expect to see in really high resolution. And we can also, and what I'll pull out of this, cumulative nitrous oxide values or emission values for a whole season. So this is the time series. So you can see kind of these different, this is just one season. So we have 6 treatments here. And so we did this essentially, this was triplicated, this graph, we have 3 of these graphs, but you can see kind of the same thing that we saw on those other graphs. So we have this peak emission period where we have the fertilizer application and then kind of a tail off at the end of the season. And then you see approximately what makes sense as far as a fertilizer application kind of dose response relationship where you see the 40% are at the low end, you see those 80% are at the high end, and the 60% are almost the same as 80%, but maybe you could do them a little lower. And so this is not a perfectly controlled system. We do have a lot of variability what actually comes out of here. And so knowing that, we're really going to talk about this kind of looking at the differences between the treatment pairs as much as possible. So kind of the headline numbers from these experiments. So we're kind of at the tail end of our 3rd season. So we'll have done 3 seasons. We haven't got the yields for that 3rd season yet because we're not to maturity yet, but we do have most of the nitrous oxide emissions. So we can do some of these analysis. So for example, this is the average change between treatment pairs. So this is just saying, okay, I had the 40% with 40% without pivot bio. What is the change for every treatment pair, and then averaging that over all the seasons. So this is kind of 0.1 or negative 0.1 plus or minus 0.3 kilograms of nitrogen per hectare. And so it's pretty similar to a pound. It's a little bit higher than what it would be for pounds of nitrogen per acre. But this is not significantly different than 0. So you can see that is a pretty high uncertainty on that, but it's not significantly different than 0. So that's kind of the big takeaway here, is that we don't really have evidence that the application of pivot bio is really adding any nitrous oxide emissions other than at the same fertilizer level. So if you apply the fertilizer level and you apply that fertilizer with pivot bio, we can't say that there should be a nitrous oxide increase with that. So, and that's what we want to be able to say. We want to say that if you apply pivot bio, if it's going to be a solution for some nitrous oxide emissions, we don't want it to contribute to nitrous oxide emissions. And so I've just kind of pulled these out a little bit here. So these are the average emissions at each treatment level over each season, or kind of aggregated, or the average for each for all 3 seasons. So you can kind of see the variability here. You can see that 40% level is almost always lower. And then there's no significant differences in any of these treatment groups between the pivot bio and the not pivot bio groups. There is kind of between the 80% and the 40%, but we expect that. So key takeaways here. So, and what this is kind of suggesting is that proven might be able to be used to reduce total N2O emissions if it's paired with fertilizer reductions. And so some of the marketing that you see from pivot bio is kind of like, oh, all our farmers put all this nitrogen and they also put proven on, and they got a few percentage higher yield. So if you're doing that, you'll still have all that fertilizer there. You're still going to have N2O emission. So, but if you can convince a user to actually reduce that total nitrogen that they're adding, you can potentially also reduce nitrous oxide emissions. So that's kind of the key point here. And so convincing them that their yields won't be significantly reduced will be the key point way. So pivot bio will have to convince us all that the actual nitrogen that they're delivering, as well as the yield results that we'll have, can convince farmers that they're not going to lose yields and lose money. So to kind of recap here, so these denitrification inhibitors, so are current, we can't really validate these at a field scale or at a lab scale. We'd very much like to. There's some promising literature there, but currently we're not able to say that that's something that we kind of have in the pipeline. Maybe in a few years we'd love to. And then these kind of tailored nitrogen fixing microbial communities, it seems like if they're paired with fertilizer reductions, there's a reasonable argument that we could reduce nitrous oxide emissions. So lastly, I'd just like to thank some other people in our group that help with this project, as well as the undergraduate mentees. And so the procyanidin work is funded by the MinDrive, and then the proven work is funded by the Minnesota Corn Growers Association. And then the facility we're actually working in was built by an NSF MRI grant. So all of these different funding sources coming together for this project. And with that, we can open it up for questions. I don't know. So first question here is, did we attempt to enhance denitrification conditions? So I think this is talking specifically about those inhibitors. So that's one potential path. So we have not, for example, fertilized soils. We were just doing kind of base soils we had on hand and looking at how they affected those base rates. So we did have soils that had relatively high denitrification activity in them, but we did not, for example, try to enhance it in those assays at all by like adding additional nitrogen before doing the assay, for example. Because the assay should add plenty of nitrogen, should be able to characterize that soil specifically. So, yeah, since it's an online question, it's hard to know if that's what they wanted to know.
I'm just kind of curious, based on the term procyanidin, if that means that it's encouraging the production of cyanide.
No, no, that's a great question. So, oh yeah, sorry. We were asked, the term procyanidin, is that encouraging the production of cyanide, which is a wonderful question. And I forgot to mention, I took some slides out, that these compounds are really well studied in a food science context. And so they're something that you'll actually often find in a supplement. So they're considered a polyphenol flavonoid that's made out of the same things you like find in green tea and different polymers. So we're not producing any toxic compounds. They're naturally sourced from a lot of fruits. But no, they don't produce cyanide. Great question though. So other questions?
So why is pivot bio getting so much traction other than making fluids? What do you guess?
I think that they would say because they're, sorry, I appreciate it. So I was asked, why is pivot bio gaining so much traction? Otherwise, other than that, they're throwing money at it. So I think that's a great point, is they do have money. They have a pretty large footprint in a pretty large laboratory and research side as well. And I think they've shown some that their product doesn't do nothing. So whether it does everything they promise, I think we'll kind of have to see. If they're really able to deliver 40 pounds of nitrogen, that's really going to have to bear it out in a lot of those field studies that aren't as controlled, and farmers are going to actually have to apply it and believe them, essentially. But I think it's partially they have money and partially that they've shown some results. So like I'm not here to tell you that they're the snake oil that's actually the panacea for all our problems either. So, but is that clear? Okay. Other questions?
Any other questions for Alexander? All right, let's give him a hand.